I'm Professor Gary Hulse. I'm Professor of Addiction Medicine at the School of Psychiatry based in Western Australia. I've been involved in this area for 40 odd years. It was difficult to know where to start in a talk about the cannabis and the cannabinoid system. I thought the easiest thing to start off with was to basically say that the CB1, CB2 receptors in our body don't simply exist so that individuals can get their hit from cannabis. Rather, they're part of a intricate system that are involved in development and cellular and organic organ maintenance. And like any other system, if we interfere with these, then we're likely to have negative outcomes occurring. This was just to remind people about the CB1 and CB2 receptors located in most organs of the body. And they carry out, as I said, maintenance function in the germ cells in our reproductive system. They carry out meiotic and normal mitotic activities. The thing that I need to reinforce about the endocannabinoid system and the way it works and the way that the exogenous cannabinoids are going to work on this is that you need to reach a certain dose before you actually affect a change, before you might start to disrupt the cellular machinery that's going on. This is something which is common to the range of cannabinoids, CBD. It's basically because there's an volatile nuclei and they have a C structure which is common to all the cannabinoids across the board. If you can disrupt this, then you're going to get cellular disruptions. We don't have the mechanisms working properly to maintain cellular function. We can get carcinogenic activities. We can have neurotoxic, immunotoxic, genotoxic. Part of the difficulty is when the issues of cannabinoids arise and we listen to what governments or companies have to say, they often tell us that there's very little information. What we really need to do is more research. In actual fact, there's a flotilla of information already out there which needs to be synthesised and collected. What they would have you believe is like Mother Hubbard going to the cupboard and finding that it's bare, and that's simply not the case. This is just to let people know that the information we're going to be providing will stimulate independent thought. That's quite a dangerous concept for many individuals because people really like to move towards conformity and to follow the social norms. I thought, how do we get we get an explanation about the role of cannabinoids and our endogenous systems? And I thought, well, we could talk about congenital abnormalities, we could talk about cancers, and I just thought, listen, let's take a simple system. Let's take the reproductive system, something which is fundamental to the basis of life, and walk you through how important what the endocannabinoids, what your naturally occurring cannabinoids do, and then say what happens if we disrupt that system. We're going to look at the integral nature of the endocannabinoid system, and then what happens if we get exposure to that, and let's look at reproductive and what happens to transgenerational effects. Just a reminder that we have CB2, CB1 receptors in our ovaries, in the corpus luteum. So the corpus luteum, this is the functional structure left after you have ovulation in the uterus, testes, prostate, and placenta. This was a seminal study which was done in 2020. And what it did was map the CB1, CB2 activity through different parts of the female reproductive path and look at what these systems normally do. It's a seminal paper. The reference is up there, and it does go to the heart of how important the cannabinoid system is, our endogenous one, to natural functioning. It actually mapped right down from the ovary right down your fallopian tube through the ampullorasmatic junction, where you get fertilisation right down to implantation in the uterus. And this is just a quick summary of that, and it's just telling us that, look, when you're getting your egg maturation, we have lower N-anandamide levels. They become high, and that's a signal for ovulation. And then when it enters into the oviduct, it's quite low when we have the sperm meeting our egg at the ampholyrismatic junction, we get fertilisation, then a really amazing thing happens. We have an increase in the level of anandamide running down the oviduct until we reach a very rich part of the uterus where there's lots of blood supply. So it basically guides that fertilised egg down there. It's the green light to come down, and then when you get to this nice rich part, it turns itself off, and that's the signal for implantation. We disrupt that by putting in exogenous opiates, then that's when we get things like ectopic pregnancies occurring. So very intricate system, and then we get changes during pregnancy, which we'll talk about a bit later on. If we're talking about introduction of cannabinoids in and how it affects the morphology of sperm, we've known for years, since the 1970s, that the acrosome cap, this is the cap on the top which allows the penetration of the sperm into the egg. It's abnormally shaped and it doesn't function properly. Its little tail on the bottom is disfigured. It can't swim in a straight line. Obviously, if you've got these problems, then you're going to have fewer people impregnated. You know, if we were rocket scientists and we had a diagram of what our sperm should look like, or the rocket should look like, and it's not looking like that, you know, clearly something's going wrong. We've got a problem here. One of the things that was also found was that the gestational exposure of males exposed. So these aren't the males who have taken, these are the ones who have been in pregnancy at the time, so they're gestationally exposed. They also have effects. So they have reduced hypothalamic pituitary gonadal hormones, reduced sperm counts, and testicular weight. So this is something being passed on to the next generation. And this is an important issue because people often talk about their right to use cannabis, apart from the fact they're not picking up the healthcare bill. But it does raise the medico-legal issue of the right of the individual to use as opposed to the right of the unborn child, which is then being impacted by that use. This group, which was exposed in pregnancy, they have different methylated regions. Now, the methylated regions means that we have, the methylation is what allows that particular part of the gene to reproduce properly on our chromosome. And there was thousands of differently methylated regions in cerebral cortex, the hippocampus, nucleus accumbens. It's important to remember this because we'll come back. What we're finding with the epidemiology, of course, is that this cellular organ disruption is leading to congenital genetic neurological abnormalities and cancers. That's very clear from the epidemiology. And this epidemiology isn't new. The epidemiology showed this with congenital abnormalities from Hawaii many years ago in cannabis users. This is another seminal study which was done, and it's terrific because what it actually, we know that the genes are involved, and it looked at the genes of people who were dependent on cannabis. And what it found is that there was a whole range of genes which had been disturbed through it. And if you look at those genes and what they're responsible for, you can match this up with the epidemiological outcome. So all this is starting, this is in humans, this is not in rats. So we know these things are going on. What was even more disturbing was that the most severely affected genes in the sperm weren't from those people using, but those who had then been withdrawn. And that's likely because if we use cannabinoids, our receptors can't distinguish between the use of cannabis and endogenous anandamides, and so it thinks we're producing too much. You drop the bottom out of your naturally occurring anandamides, and then you take the cannabis away, and we've got no signaling going on there at all, and this was a real problem. So this was the Murphy and Scroll research groups. They showed there was changes in methylation over thousands of different regions to do it. And again, if you match up those, it relates to the epidemiological data of human development, congenital and genetic abnormalities that we're seeing. Should we be disturbed about this? Well, clearly we should be. This is something that really concerns me. This science that exists out there, people are often saying we need more information, we need more science, but the science is already sitting out there, and yet We often have the monkeys, the see no evil, hear no evil, speak no evil. We have a third one sitting on there that seems to be protecting their reproductive organs themselves. And so you can make this person who you want, perhaps politicians or others. But science is being ignored, and we need to garnish this science together and look. Those people who raise a voice, perhaps like ourselves, are often people want to silence you. We're raising issues of caution. There are probably three takeaway messages that I'd like to give. One is that we know that we have a biological clock. We know that there are certain cellular manifestations that we can use to measure the biological age of cells. And we know that people who use cannabis age more quickly, and therefore they get age-related disorders more quickly. This is unequivocal. We already know these things. It does it by unraveling and disrupting the mitotic and meiotic cellular reproduction. It exerts the genotoxic and the disruptions across the range of cannabinoids, CBD as well, because they all share this overtol nucleus, which has a C ring on it. And this is the component. Has this just been discovered? No, it was discovered back in 1977, and yet it's a piece of information that's been confirmed many times since then, but a piece of information which tends to be forgotten and dropped out. And like many substances which form homeostatic balances in our system, we need to reach a threshold before we have a switch-like effect where we get these effects occurring. And this is really important because some jurisdictions have at the moment gone down the path of decriminalisation of cannabinoids. Places like Colombia, Canada would tell us that they haven't increased the prevalence, the number of people using, nor the frequency, which might all sound very good. But if what we're doing is allowing companies to continually increase the amount of cannabis in each of the products we're going through, then we're reaching towards these thresholds, and these switch-like effects are going to come into action, and we are going to get congenital, and we are going to get cancers. And this is exactly what we're starting to see in the epidemiological data coming out. So with deregulation, genie already out of the bottle, for the sake of not only the current generation but also the future generations, the time to act is now. And it's a plea. We really do need to look at the existing science, take it seriously, and let it direct political decision making rather than commercialisation. Thank you.
I'd like to acknowledge my collaborators, Professor Gary Lynch and Maurice Sim and Lorinda Lynch. And we're going to talk about cannabinoid neurotoxicity and genotoxicity today, talk about mental illness, autism, cancer, congenital anomalies, and aging. The 5 alphabets of life are disrupted by cannabis. The genomic code, 59% of the chromosomes, the epigenomic code, 20,000 hits, the histone code, the tubulin code, and the sugar code, all disrupted by cannabis. Now, the brain matures, as you're probably aware, till the mid-20s, and this is a picture of synaptic pruning going on and maturation. This is a picture of tobacco, alcohol, cannabis, and cocaine across the US. And you can see that the cannabis is concentrated in the northeast and northwest and centrally in Colorado. When you look at the mental illness, any mental illness, depressive illness, serious illness, suicidality, again, it's this northeast, northwest, central pattern. And you can show statistically those things are correlated. In most US states, the rate of autism is rising exponentially, and you can see there's a map of it across the US. Now, if you look at the high cannabis use states, the low cannabis use states are 61% more in the high cannabis use states. Now, if you look at cancer, this is on the x-axis is tobacco use rising here, and on the y-axis is cancer rate rising. And you can see for these 14 cancers, it's obviously positive. So you've got lung cancer, laryngeal cancer, all cancer, leukemia, esophageal cancer, cervical cancer. These cancers are known to be cannabis linked. Same thing for alcohol here. Again, there's 14 cancers there for alcohol. Look at last month cannabis. Again, here there's 16 cancers obviously positively trending with cannabis. And if you look at daily cannabis, the same sort of picture appears. This is the THC concentration of cannabis herb. And you can see there's about 20 cancers here, positively trending with rising herb THC. And for resin content of THC, again, it's a similar pattern. Now, this is a very interesting map of Europe. This is the rising rate of THC in the herb, cannabis herb, and the rising rate of breast cancer, so that the two rise together, both covariates rise together. And as you can see here on the color plane, that the whole continent's turning pink. It's a very dramatic appearance there of cannabis and breast cancer rising together. For pancreatic cancer, it's a similar pattern. Here, the whole continent's turning purple, which has a similar meaning to the pink, as you can see up here on the color plane, where the whole continent turning purple as both cannabis and THC herb rise together. And if you look at the cancers which are more common in the high cannabis use countries, you can see that it's much more common in science. P less than 0.05 is significant, and here it's P is less than 10 to the minus 135. So it's much higher in the cannabis using high cannabis countries. And here is the number of cancers implicated by a substance. The total P value is the significance, and this is the E value, which is causality and logic and confounding. Much higher here in each case, number of cancers, P values and E values, much higher for cannabis, and tobacco and alcohol aren't rating there. Let's look at congenital anomalies. This is a blue baby from cyanotic congenital heart disease. The guts are hanging out here, gastroschisis, a dead bowel, dead baby. This is no limbs. 2 papers here from CDC showing that limblessness is linked with cannabis, and also in Europe and America we found that. And carefully, this is 2 papers from America looking at no brain, and Europe showing the same thing, no brain. And carefully here, holoprosencephaly, severe facial brain defects. This is tobacco. This is slide 1. So here we have the tobacco rate. And here's the various congenital anomalies. And you can see here, there's about 20 congenital anomalies positive related to tobacco. This is the rest of the 90 defects that are tracked in Europe. This is last month cannabis. And most of these are positive to some extent, much more than tobacco. And here's the second slide of that. Daily cannabis use, again, even more strongly positive. There's about 50 or 60 defects linked to daily cannabis use here. The teratogenic syndromes, so you can see this is Belgium here, is strongly positive. Germany here, Bulgaria here are the positive countries. And on the bivariate map, you can see Spain and France are turning purple here as both rise together. There's Netherlands positive there for teratogenic syndromes and THC herb. A VACTAL syndrome, vertebral, anal, cardiac, tracheoesophageal, renal, and limb are rising together. That's Germany, France, Netherlands. Right here, you see black, and on the bivariate map, you see the low countries are pink, and purple is both VACTAL syndrome. All these 6 systems and this metric of cannabis rise together. Now, this is the 9 systems involved. You can see all these 9 systems are involved. And this is comparing cannabis here and tobacco and alcohol here. This is a number of congenital anomalies, the significance value, the P value here, and the E value here. And tobacco and alcohol aren't rating here, and they're much lower here on the P values, and the number of anomalies is less. Let's talk about chromosomes and some of the cellular mechanisms which produce this. Now, this is a developing notochord, the spinal cord in utero, bone morphogenetic protein and the Wnt proteins here. You can see there's gradients of these morphogens which control sonic hedgehog ventrally, bone morphogenetic Wnt dorsally, at the back of the developing embryo. This is the spinal cord. You can see these morphogen gradients specify exactly where each of these cells is going to lie, and cannabis blocks the gradients for most of these morphogens directly and epigenetically. This is human sperm, and you can see here there's a normal sperm, look like tadpoles, but on cannabis, the head is shrunken. This sperm's got 4 heads, that one's got 3 heads. They've got bent tails. Some of these sperm have 4 tails, multiple heads, shriveled up heads here, and a pus cell right there in the middle. Same thing here with the egg. This is an egg dividing. That's a chromosomal bridge forming. That's severe genetic damage, a snapshot of it actually forming. That's also an aging sign. And here you can see a cell dividing into 4. Here's one dividing into 5, and here's one dividing into 3. Grossly disorganized cell division here. And in fact, 20% of these cells die with just a single cell division. Same thing here in the mouse sperm. Here you've got a ring chromosome, there a chain chromosome. Here's chromosome pulverization and microchromosomes, all forming as severe chromosomal disruption. Now, when you add up all the defects, the birth defects that cannabis involved in, and the cancers, you find all these chromosomes are implicated in direct genetic damage, which is 1,765 megabases out of 3,000 megabases in the whole human genome. 59% of the humans directly involved, known to be directly involved in cannabis genotoxicity. Let's talk about aging for a minute. These 20 hallmarks of aging are all accelerated in cannabis. They've got an old sperm, an old egg, and obviously the fertilized zygote is old, even from the moment of conception. It's aged from the moment of conception. That's a mind-blowing concept. Now you see these 20 indices of aging. The genes, the DNA carries the switches on, the epigenetic switches, which turn the genes on and off. As you can see here, these methylation marks, which turn genes on and off and control which genes are on and which aren't. So this is cannabis in cannabis dependence. This is an epigenome-wide screen. This is brain development here and here. Agenesis of the whole organism doesn't grow. Growth of the organism, heart development, liver lesions. This is cannabis dependence. When you move to cannabis withdrawal, you've got the hippocampus growing here, the brain growing here. This is the blood vessels growing here. Organismal death, the death of the whole organism. Learning and cognition here, which is thinking. And encephalopathies, brain development. And down here is the testis, is most commonly involved. So all these systems. Now this is a very interesting, just published study of aging to show us why we age. There's a loss of epigenetic information in aging, which drives aging. So that the activating marks that activate genes here, see how they're reduced from old to young, and the suppression marks that turn the genes off are also reduced from old to young, which reduces the control of the genes. This is enhancers, which turn on the promoters. And here it's nice and strict with a strong epigenetic landscape in the young, which is all eroded and all messed up. We've got a major loss of information here in the old cells. Now, this is the basic epigenetic machinery, which is shown to be disrupted and destroyed by cannabis, so that the fundamental software that controls gene expression is destroyed by cannabis. This is a genome-epigenome-wide correlation showing that for brain development here and congenital anomalies, birth defect, cancer, from 90% here to 25% of all the genes. This is the genes involved with cannabis. This is the total genes implicated. And you can see there's huge numbers of genes, thousands of genes involved in direct cannabis damage. And of course, the death rate is much higher, 30% here over 30 years in cannabis use. Now, an important point here is that a lot of these effects are exponential. So as the concentration rises, this is mutagenicity and damage to the cell. Here, you can see it rising exponentially or falling exponentially here with cell growth. And here again, with developing red cells, erythroblasts, you see exponential as the concentration rises, exponential effect with gene damage. And this here is the same thing observed with thalidomide. This exponential effect as it hits a point in the concentration rise and takes off. Now, just looking at thalidomide is known to cause 31 birth defects, and 21 of those are also noted with cannabis. Thalidomide acts in 13 different ways, and of those mechanisms, cannabis shares 12. So 90% of the ways that cannabis does it are also involved. Many cannabinoids are involved: delta-8, 9, and 10 THC, cannabidiol, cannabigerol, cannabichromin, and cannabinol. So the 5 alphabets of life are disrupted by cannabis: the genomic code, the epigenomic code, the histone and tubulin code, and the sugar code. Talked about mental illness, autism, cancer, congenital anomalies, and aging, epigenetic, transgenerational, exponential effects. Many cannabinoids are involved. Thank you for your attention.
Thank you for attending. It's great to see everybody here, and we're really at a moment. It's been an important conference, and I think that we would all agree that this topic is something that's really very important for us to focus further on. Now, we've just heard a number of presentations that I think you will agree are really new information. A lot of this has been published just only in the last 3 years. So I think that you shouldn't be surprised if what you're hearing about today is something that you hadn't heard about before. It is some information that I would suggest is a game changer. And I do think that it puts us in a very different world now that we are understanding this body of information, where we see that you've got, for the first time, many of the databases that look at congenital anomalies across the USA and different states, and they've been organised in a time series and linked to this body of information on patterns of use. And we see here a clear signal that both the congenital anomalies, the birth defects, and the cancers are very much causally linked. This is a whole population level. So very concerning, and we see the same pattern when the delving into further papers that are related to Europe. Now, Gary has explained very carefully that this is not a new finding, that this finding of genotoxicity is something that has been shown through the science over a number of decades. Now, what I'm really going to focus on here is the issue of how cannabis legalization is actually feeding into this situation. I think we're at a moment, it's a moment where we really do need to take a good hard look at what's occurring here. And I think that we would agree that what is occurring here is unacceptable harm. My background, I'm the professor and the chair in health psychology at the Australian university called Deakin University. You can see here that our research group, the Centre for Social and Early Emotional Development at Deakin, is very much concerned with looking at the body of data around longitudinal life course studies. So you'll hear about some of the studies that we've actually gathered data from. We're collecting data from a number of the Australian and New Zealand cohorts and linking them together, giving us unprecedented access to the longitudinal picture. We're also very interested in what works for interventions, and today we're talking about what we might do to make a difference across very large populations. So in summary, what I'm going to be talking to you about is I'm very much wanting us to be on the same page as to what the evidence is saying. And so rather than just hear from me and my selections of studies, what I'm going to present to you is what we're getting in the signal from the systematic reviews that have been recently published. And we'll have a look at those. We're also going to have a look at evidence from longitudinal studies. And I'm going to share with you some of those that I've conducted and my team have conducted. Really, in a nutshell, what I'm saying here is that cannabis legalization is increasing availability and social acceptability of cannabis, which is increasing both use and harms. And I'll argue to you that there is a very strong signal that cannabis use is related to genotoxicity, is reflected in birth defects and cancers. And so I don't think I need to argue with you that this is a bad situation that we're in. This is a moment where we really need to pause and change what we're doing. So I'm going to present to you some potential ways forward if we take a rational approach of using the prevention sciences and to do those things that we've done successfully in the past. So we know we can be successful with prevention. Here we are, we've had massive changes around tobacco use, and you've heard how Shane has presented the way that tobacco was once something that we were sort of told was very acceptable, but no longer is. And the same is also true with alcohol. We once thought that it was acceptable in America to reduce the drinking age from 21 and take it down to 18. Lots of states went along that pathway. They then found that they had increased road fatalities, and there was a reversal in many of the states, and they rose the drinking age from 18 to 21, with the effect that, again, they saw changes there with road fatalities coming down. That led to a consistent approach across the US to try and reduce the road fatalities by bringing an age 21 drinking law. So the idea of actually using policy to try and improve public health is not new to us. We've applied it many times, and we can do it again successfully here. So when we look at the evidence from systematic reviews, we see here that it is possible to do a review very quickly. I've done here using the PubMed system, and you can see using these terms, I was able to identify 107 papers over the past year that have actually been review articles. Of those, there were 5 that actually listed systematic review of the topic we're looking at in their titles. And when I look at those papers, I find 4 out of 5 of them found evidence of increased use and or harms that are associated with legalization. So I'm very keen to try to make sure that, as I said earlier, that we're on the same page in terms of understanding the evidence base. And so the way that I've approached this is using this method, which I think we would agree is actually a method that we can come to some consensus about what's going on. And it is sending a fairly clear signal that our current process of changing policy around cannabis isn't really doing the things that we might want if we aim for public health. It's actually taking us in the wrong direction. So let's look at these studies. So this one is by Windle and colleagues, and we can see here that they have found 64 studies. And of those studies, changes that they saw in the US and Canada associated with road impacts. We see here that both with medical and recreational legalization, There have been some increased use among drivers. We see here that with the medical legalisation, there was a reduction initially in fatal motor vehicle collisions. Well, that's great. We would love if policy could actually reduce problems, and we will celebrate that when that is the case. But unfortunately, with the recreational legalisation, we've seen increased fatal collisions. Here, the Chance study with 32 papers included looking at youth consumption associated with legalisation in non-medical cases for Canada. There was actually an increase in adolescent past month consumption, and there were some increases amongst young adults. You see in 42% of the quality studies. So while there's some mixed evidence, we do see a signal coming through here. And in 33 studies here by O'Grady and colleagues, looking at non-medical legalisation and their effects on adolescent and young adult consumption. And then what we saw is for that recreational legalisation, increased young adult cannabis use, not a good thing, you'll agree. And then with the results for adolescents in that study mixed. For French and colleagues with 113 papers summarised, here we see legalisation increasing adolescent cannabis use and decreasing adolescent perceptions of harms. Again, those social norms are degrading, with young people being led to the wrong conclusion, thinking that cannabis use is safe and harmless. Here we saw reduced educational achievement in 3 of the 3 studies that have looked at it. However, these economists didn't find yet evidence that they could get a clear evidence of a negative social cost. And so it may be that there's longer in this huge social experiments that we're undergoing before that comes to fruition. Here in this study, we see 32 papers reviews looking at the non-medical legalisation in the US. And here we see increased adult use and young adult use with the adolescents not showing the same signal. Here, preliminary trends are evidence with increased service use and traffic fatalities, as we mentioned earlier, and potential increases in serious violent crime. So in summary, what we're really seeing from legalisation from the systematic reviews over the last year is increased use by adults, increased adolescent use in some studies, more hospitalisations, more cannabis use on the roads, and potentially with at least one study showing more crime. But societal costs are not yet higher. But I think we would agree, even if cannabis use was not increasing and it was staying the same, that would not be good policy in the current situation where we're showing you very clear evidence of these genotoxic effects. The results we saw from the recent reviews reflect those in this study by Fisher, which was one of the previous reviews, and you can see very much the same sort of signal coming through with this set of findings. I'm going to now present you some findings from longitudinal studies, and I'm going to first show you a study that we published in 2014 where we brought together the Australian, New Zealand cohorts and looked at the effects of cannabis use in young adults where adolescents had used cannabis. And here we got a very clear signal across multiple studies showing that that use in adolescents was associated with dependence, illicit drug use, increased welfare, increased mental health problems. And this is really where we've been for many years very concerned about these types of findings that characterise much of the previous research. We were told, no, they will not be the same under legalisation. So here what I'm showing you is a result of a cross-national comparison between our state of Victoria and Washington state in the US. You can see the aims of this study here. There were 3 aims trying to look at these comparative effects on behaviour. You can see a very good study. The state representative samples very large, with over 73% participating of the recruited sample in both states, and terrific follow-up rates that you can see listed here. With legalisation, there was the introduction of the recreational cannabis stores in Washington state from 2014, allowing us a natural experiment to compare how young adults develop in these contexts. What we saw immediately was higher cannabis persistence and uptake in Washington state, and you can see the figures here, with 17.5% persisting in Washington state compared to 6.4% in Victoria, and double the rates of uptake in Washington state. So concerning figures in an environment where, as from this transition from 25 to 29, there is actually conception issues that emerge. So here we see, when we looked at the issue of whether or not the legalisation could have maybe reduced some of the harms, what we did find is that actually availability, prevalence and acceptability were all rising in Washington state over that period, and they were equal contributors to use at age 29. So concerning again that these things are quite known to us as influences on use, and we've known that from the literature on tobacco and alcohol. We went and further examined whether or not harms might reduce in the legal environment, and here we see Victoria and Washington compared for the consequences that occurred in age 29 in 2018 for using in the past 30 days at age 25. Here we see that actually cannabis use at age 29 was more likely to occur if you had used it at age 25, and there were no differences, non-significant differences between the two states. For cannabis problems, here we did see a state difference with slightly fewer in Washington state compared to Victoria, but overall cannabis use at age 25 was contributing to greater problems at age 29. Here we see clear no differences despite the legal policy in cannabis use leading to cigarette use with a signal in both states and leading to opioid use. We see with marijuana driving that there was actually increased risk of that at age 29 in Washington state despite the legal policy. Here we see depressive symptoms and suicidal thoughts both increasing if you had used at age 25 with no differences between the states. Here we see self-harm showing a state difference and no difference in the sleep problems. For these other ones that show a signal that there was homelessness is increased through the earlier age use. Here we have interesting findings with financial difficulties and low income risks are increased. Again, very much in line with our earlier finding prior to legalisation that the adolescent cannabis use was increasing welfare. It's often argued that these policies might reduce trouble with the law. Here we actually see that incarcerations are higher in Washington state compared to Victoria, and court offences are exactly the same despite the legal policy. So here we find that cannabis legalisation is very much increasing established risk factors for use, availability, perceived prevalence and acceptability, leading to increased young adult use and very similar long-term trends in harm. So I guess that if we say that this was a healthy policy, we'd have to argue that it wasn't. So there's really, in summing up, what should we do about this situation where we really do need to change course? The first thing to say is that it is possible to introduce healthy policies. The first thing to agree is there's really no safe level of cannabis use with adolescents and young adults. We need to understand that. Cannabis legalisation is increasing established risk factors for use and all potential harms that have been discussed in this seminar. So really what we're arguing today, this is the day where we really send out very strongly the call to arms that we've got to step up prevention advocacy. The findings we have are a game changer, and we really need to start to do the things that were successful in tobacco control. We need to regulate markets using similar effective tobacco control measures that we have seen previously work to stop these growing harms. So I thank you for listening.